but I'm here for you, my friend. In troubled times we are apart, but I'm here for you, my friend. We used to sit in the park till dark. I love those days outside with you. I'll always remember you by my side. We're never really far apart. Life is hard as we know it, but I'm here for you, my friend. In troubled times we are apart, but I'm here for you, my friend. Remember now to smile, remember now to laugh. It won't be long till we meet again. Remember now to smile, it will only be a while. Soon we will meet again. Life is hard as we know it, but I'm here for you, my friend.
How's everybody doing? Hopefully things are going well. I am still working on some student projects, and this is sort of an interesting side project that came up relatively recently. This part is not part of the student projects, but since I was going ahead and looking at stuff in the scanning electron microscope today, I thought I would give it a shot and see what I could find. It's a relatively short process of what I'm doing. Really, I just was checking through the sample to see if there were any diatoms in it. What's interesting about it, there are diatoms, as you can see. That's one of the diatoms there. What's interesting about it is when I zoom out, you'll see we are actually on the tulsin, which is the tail of a crayfish, a crawdad, if you like. And just one of the little plates on the tulsin, and it was covered with a little bit of brownish orange stuff, and I was relatively confident that it was algae, and I thought that it may actually be diatoms. And people who've been following the channel for a while probably know that I'm kind of interested in diatoms growing on weird things, like sloths and hippopotami, and I'm also kind of interested in other random things that they might be growing on, and this is a diatom here. I don't actually know what the species is. It's not prepped for diatom analysis, so some of the organic stuff is still on it. So that's part of the issue, but probably if we were going to scrape the sample and analyze them for diatoms, we could. So this is a diatom. Here, it's at the right size and everything, and has clearly got a raphe, so it's a benthic diatom. And they would live an attached lifestyle. And I'm just zooming out so you can see. Here's some more. There's one, same thing probably as this one. Here's some diatom. There's one. That might be a diatom, I'm not sure. That's a diatom for sure. And here's a bunch down here as well. These are clearly diatoms. So there is here's a broken one. So there is clearly diatoms living on this crayfish. And this is Cockanice. Actually can tell what genus this is. So they usually live on macrophytes, but it turns out they apparently also live on crayfish. And so this is just kind of like a curiosity for me. Part of the reason that I'm interested in it is one of the grad students here, Kathy, who's a PhD student, has been collecting crayfish from the Wabash River and surrounding river systems. And so she has a whole bunch of these. And that means I have a whole bunch of samples that potentially have diatoms on them. And they've already analyzed them for what they need them for. So they're basically just going to waste. And here's a diatom, and here's another one. So a bunch of little diatoms that live attached lifestyles, which is basically what I would expect in this scenario. And then these bristles are part of the actual tail fin of the crayfish. And this is the far end of the crayfish's tail fin. So, you know, it's covered with sort of like these long bristly spine things that, you know, we're at such a small scale, you wouldn't be able to see these very easily, although they would look like it has sort of like a fuzzy edge to it, or a hairy edge to the backside of the fin. And so that's what we're looking at here. And really, I was just sort of curious, like, are there diatoms living on it? And then, you know, the crayfish are being stored they're dead, they're being stored in alcohol. But the exoskeleton, time to get out of bed. Hello, Chook. It's early there in California. The crayfish are dead, but there's a whole bunch of them. So this is just one tail fin, and it definitely has diatoms on it. So the question is, for me, is there an easy or effective way of extracting the diatoms that are growing on this crayfish tail? And presumably the other sort of orange and brown spots that I saw, you can see it's kind of broken, but there's the whole, it's just one little tail plate, and it had tons of diatoms in these sort of fibrous areas. And there may be some actually attached to some of these areas. I didn't look carefully through here to see. I mean, this is This is kind of the scale where this is probably bacteria, but this is kind of the scale where they occur in the 50 to 100 microns. We should be able to see any diatoms that are on them. In terms of scale, but I think what we need to do is maybe process some of them, try dissolving the whole thing in acid, or the other alternative would be to scrape some of it using a piece of metal implement to try to get some off. They're not a fish. Crayfish are like tiny lobsters, basically. I don't know why they're called crayfish. Where I grew up, they were either called crayfish or crawfish, which is the way that people would say it, or they would call them crawdads. They're definitely not a fish. They're an arthropod. It's like a lobster, basically, a very tiny lobster. What it implicates, though, is that there probably are diatoms living on lobsters as well. And anything that has sort of like a hard shell. Like, we know that diatoms live on turtles. I don't know if people have looked at crayfish very closely before, but they clearly are growing on these crayfish, and they're in an attached life state. So it's not like they're just stuck on there. They're actually, in life position. So that's kind of interesting, and it means that it may be a project that I put somebody on for this summer. I have a bunch of samples from the Wabash River, and they're collecting some more crayfish. Mostly they were looking at these for mercury content, because mercury usually is a bioaccumulating metal for some organisms. And I think what they were trying to figure out is, if we look at organisms that accumulate mercury, for example, we might be able to use that to figure out what the mercury levels are like in the place where the organism lived. So it's a way of linking the environment to potential environmental problems. So this is kind of cool for me, and I'm not sure how anybody else feels about it, but I rather like it when I find diatoms in weird places. And somebody's probably sampled this type of system before. I'm gonna get the door real quick. Hang on. sorry about that. Alright, let's take one picture with some of the diatoms visible here, all in their life position, and doesn't need to be the greatest photo in the world. I just need to be able to see that there's clearly diatoms present, which I think we can see here. There's one obvious right there. There's a whole bunch of them right here. And just document that we have it. And then I'm going to jump over and take some pictures for the other student project that I'm working on. And probably just a real short stream. I'm just collecting images so that we have them for student projects that we're working on for my class. So this picture isn't related, it's something different, like potential research avenue in the future, looking at crayfish and across the distribution of places in the Wabash River. And then we'll probably try to find a way to digest these, either with acid, or I might be able to clean them, as I mentioned, and then we can just process what gets cleaned. Oh, that's awesome. It's they're little tiny, those are little tiny diplonis, I believe. That's really, really bizarre. I might zoom in on that other one and take a close-up picture of it. So I think these belong to the genus Diplonis at the top. Not all of them. There's some Cockanies in here as well. There's a Diplonis for you. Yeah. So that's this guy right here. That definitely looks like a Diplonis. This also looks like Diplonis. Wasn't totally clear to me from where we were what they were, but it looks like there's a whole cluster of little Diplonis living in here. And this one as well, Diplonis. I think this one we looked at, it was actually Cockanies down here, the broken one. So, but that one's very clear, and so I can zoom in and take a look at it. These are all pretty small. So the length of that's maybe like 25 microns or less. Yeah, that's a Cockanies. It's broken, but these ones all look like they're definitely in place. All the same species. And they seem to have made a home for themselves in this tail of a crayfish, which is pretty cool. Alright, that picture is done. This is actually from the Wabash River. Crayfish. And let's take a look at this one that looks kind of clear up here. And see if we can actually get... It's a pretty good resolution. Okay, let's see what I can do with this. Hey Moosey, how's it going? We're in a really strange place, looking at a really strange thing. It's actually broken a little bit. I'm doing good. It's the end of the semester. It's finals week, and the students are mostly not around. Samsung, hello. Working from home still. Good to see. We're looking at a little tiny Diplonese. It looks like it's maybe 20 to 23 microns, something like that. And it is living attached to the tail fin, the tulsin of a crayfish. And I did not know diatoms live attached to crayfish. I mean, I assume they're attached to all kinds of things, but I mean, these ones seem to have a preference for crayfish in that there's a whole colony of this thing. So maybe it just got here and then it, started growing and did well. But I think it would be really interesting to try to explore what diatoms, or whether these diatoms specifically occur more frequently on crayfish. Because that's super, super weird as a place for them to be. The student who gave me this material was looking at the crayfish, and they just said, "Oh, it looks like it's got some algae on it. Do you want to see if there's anything in there?" And I was like, maybe diatoms are living on crayfish. But I think this is proof that they are definitely living on crayfish. And I need to do a little bit of research to see if anybody's really looked closely at what lives on crayfish. But this is clearly a Diplonese. There's no doubt that's a diatom. And I can put it into the right genus. And with a little bit of work, I might be able to figure out what species it is. Provided that it's an existing species. But really, it was like a maybe they're there. I mean, it's brownish looking algae, so likely it's diatoms. And then these are just from the tail fin. So the tail fin, like crayfish have like lobster tails, basically the tulsin structure. And but there was a bunch of, it looked like maybe algae on the chest of the crayfish, especially around its mouth area. And so it's an avenue for me to just basically explore. It's a weird setting, and the diatoms may be pretty basic. I don't know, they might be pretty common. This is a Diplonese. It has really weird little dots out here on the outside edge, but maybe that's common for the species, I don't know. But Diplonese are raphid diatoms, so here's a raphy running down the middle of the valve in this sort of central sternum, big central nodule. It's got these like separated areolae that are around the outside edge of the raphy, and then there's a hyaline area, and that's really common for Diplonese. It creates this sort of H-like shaped structure here in the middle. So I don't know what species this is, and I don't have a book of Diplonese in front of me that I would like to look at, but it's interesting where it is. And the fact that there's a bunch of them, which suggests that they're definitely in life position. Fortunately for me, I have a friend who's an expert in Diplonese, and probably has looked at some weird settings like this before. Yeah, it's neat. So, oh no, diatoms are around today. Some of the diatoms that I looked at before are fossil. We've looked at stuff that's diatoms that are 10 million years old, and maybe some older stuff even here in the channel on stream before. Is this also a Diplanese? Yes. So that's also Diplanese. I think it's the same species. So I was looking for those little... I don't know that we'll see them, those little holes that were on the side. But these weren't processed for diatoms in any way. The samples are just... I let it dry out from the ethanol and then stuffed it in to the SEM. So, yeah, so no, diatoms are around today. They're responsible for most of the oxygen that you're breathing. Like a fairly large fraction of oxygen on planet Earth comes from diatoms. So they're definitely living algae. These are all Diplanese. It's like a monoculture. Diplanese. It's the same species. It's like it landed on the Tulsan and then basically it did very well. Or maybe this just is an area that it thrives in. That is also Diplanese. Everything's the same genus, and as far as I can tell, it's the same species. As those little dots are on the outside edge right here again. So we should be able to use that to figure out what we're looking at. And I did see some other things in here, but like these things are all Diplanese, all in this little area. And I did see a Cockanies in here, so... It was probably just the specific type we were looking at that day, Lucy. You know, I've shown living diatoms on stream before in the evening microscope, like live microscope stuff, and Pacific Plankton also, who I think you're sometimes in her channel. She's also had live diatoms on there, and our friend Diatoms, D-I-E-T, Diatoms, also looks at live diatoms pretty frequently. Hey Science, how's it going? We're looking at some really weird setting, and I found some of my friends, some of my normal diatom friends here. This is a diatom that's not Diplanese. There's a Nitzia. So, I mean, there's plenty of diatoms present here. It's dominantly Diplanese, but really there's a wide range. This is, if I zoom out, it might be clearer what we're looking at, but this is a fragment, just one of the tail fins, or Tulsan fins, of a crayfish. So this is what the crayfish Tulsan looks like in SEM. You know, at the end of it, it has this feathery bristles, and there's like a spine here I didn't notice before. But, you know, on the tail of crayfish that they use to get around, so they're swimming fins. This is just one of the plates, so the smallest plate I could find, actually, that I could pull off. It's probably charging a little bit, but it may be, like when we were up here, it may just be I need to change the brightness and contrast settings a little bit, but all of the, because I was zoomed in and now I'm zoomed out, so here, let me see if I can fix it a little bit. Yeah, so when you have long spiny things, they usually tend to charge a little bit, so. Yeah, that's okay. Don't worry about it, Moosey. So yeah, this is just one of those plates, and we were zoomed in just looking at this little furry bit right in here, and it's covered with diatoms. I mean, not totally covered with diatoms, but they're present all over. And I saw some in these as well, in the little bristly parts. So anywhere they have these sort of bristly little spines or pieces, there's some diatoms that were present in them, and I didn't see any on the flat bits in here. I was kind of expecting them to be kind of encrusting on some of the surfaces as well, but they seem to specialize in, oh, maybe there's one in here. The surfaces are covered mostly with bacteria, and then there's diatoms in there. So there's one, that's a Diplonice again, and here's something. Looks like maybe a Cockanice. Yep, that's a Cockanice. And some fragments. There's a diatom. I'm not sure what that is. It's very little. There's a broken piece of a diatom. So none of them are very large. They're all little guys. And I'd expect that that's kind of normal. And most of the ones that we're seeing they live an attached lifestyle. Here's a little diatom just tucked away on the inside of this, right inside here. Yep. I don't know which genus this is. Hang on. No, it's just a Navicula, a regular old Navicula. I don't know the species, but that's a Navicula. Very flat one. Very small. It's 10 microns basically across the length of it, maybe 15. So, drone hands. Hey, hello, drone hands. Drone Hands is a musician here on Twitch. They're a streamer, and they do really cool electronic music, and they're working on an album. And our friend Studio Cornix really likes them, and rated them a couple of times. And I was hanging out with her. I really liked it too. I don't listen to a lot of electronic music, but I thought it was really well done. So, and I think you should give them check them out if you like music. And the streams are pretty chill. Just kind of hang out, playing stuff, and conceptualizing music, I think. So, what are the largest diatoms? So we had some. There's some really long, skinny ones that we've had, that we saw on some samples from Pacific Plankton, where it was like maybe 2 millimeters or something, like really big for a diatom, but long and skinny, right? So not very massive, just like this, very like a hair basically. The ones that actually have like a rounded shape, like that have like some robust quality to them, a couple of millimeters is probably the biggest. They're marine, so I don't usually work with the marine ones. And then in the freshwater realm, it's probably much smaller than that. It's probably like 400 microns or something. I don't know. I don't see much that's bigger than that in freshwater systems, but in the marine systems, because it's got a little bit more wave energy and high diversity of plankton, there's probably some bigger stuff. So pretty cool that there's single cells that are that big, though. Okay, anyway, so this was just a fun little, I wanted to see what was there. This is not part of the student projects. It's potentially a student project for the future. Also, I wanted to mention we have some student-supported research that's going on, where I'm using the money from subscriptions and from Twitch or whatever to help fund student projects. And one of those projects is looking at this stuff from June Lake that my undergraduate student Daphne's working on, and we wrote a little grant to try to get her some money for it to go along with the money that we're getting from Twitch. And we got a little $5,000 grant that was funded yesterday, we found out. So that's pretty cool. And she still doesn't know, so I haven't told her yet. But those aren't silly questions. Evan, hello. I'm pretty sure you have an automatic shout out if you talk, Evan. Yeah, the pictures that are down below or on the right-hand side of the black and white ones are, there you go, Evan. Welcome in. And yeah, it's just a little tiny grant, but it's always nice to get something funded, and it doesn't really require a lot of money. And thank you for the subscription as well, Evan. So that project will be very well funded, which is nice, and I'll be able to pay for the student to do the research for next fall, probably exclusively, maybe for the whole next year, really, exclusively from that grant. And then there's a Diatom meeting that's coming up in September that I'm hosting here at Indiana State, not on campus, but in the field station. And I'm hoping to have a bunch of my students able to actually present their research there at that meeting. So that'll be really nice, because you'll have some extra stuff to talk about. Hey Luke, what's the hairy thing? The hairy thing is a piece of a crayfish tail, the Tulsin fin, and they use those for swimming. To answer your question, Drone Hands, the colored image are all colorized in like Photoshop, basically. Those are art. Those are science art that I created from the images. So I took the picture and then I took it into Photoshop and I manipulated them to add color and to try to be creative, or to pick out features that I was trying to emphasize. And then I post them to Instagram and my Discord and Twitter and things like that. I think that they help draw a lot of attention. Like if I just posted SEM images that were black and white, people probably would still like them, but it seems like the color kind of captivates people a little bit more. And so I've been colorizing images for almost since we started taking pictures here on this SEM, as a way to, sort of broaden the science outreach for Diatoms and the research that I do. So it's I actually have some people who kind of resist it. They wanted to see things in black and white, or they feel like maybe I shouldn't be colorizing things because it confuses people. But I don't think it actually takes that much to explain, like, we colorized it. It was black and white image. The reason for that is, for people who are curious, diatoms don't have a color for one. Well, especially after they've been processed. They are, once they've been processed, you're just seeing the skeleton, and it's clear. They're like glass, basically. And so there wouldn't be any color to them other than physical color, like from light reflecting around in there. But additionally, the scanning electron microscope doesn't use light. It uses electrons to see. So everything that you're seeing has been created by having little electrons scan over the surface. And as a result, we can't have color images because we're outside of the range of color wavelengths, well outside of it. We're in the wavelength of electrons, which is, angstrom size. So well below the nanometers that you use for light. So we're not in the light color range, so obviously we can't use light. But I think it's fun to colorize them and just post them. So they're here as images, and they're on Instagram and whatever else. So that's science art. The images by themselves are pretty stunning, I think, but I'm a scientist, so I have kind of like different objectives when I'm trying to see. So everything's going well, Evan. Thank you. What's up, Cyanophyte? Hello. And Procyon? Hello. Look at that automatic shout out going for Cyanophyte as well. It's ethically wonderful. Yeah, well, that's what my objectives usually are, is to try to... I'm an ambassador for a bunch of invisible dust-sized algae. So I try to do my best to make people aware of them and enhance people's recognition of them, so that when they see it, they go, oh, that's a diatom. This particular diatom is a Discostella, and this is a diatom that we wrote a paper about. This is an invasive species, or non-native species, or at least we think it is. This is Astera costata, and it looks like a little spaceship with these little tiny Mantophotoportula, the little openings that stand out on the surface. I can tell it's a Discostella because if you look at these solid lines that are here, these are what we call costae, or ribs, on the valve, and they're on the edge of the valve. And if you look at the position of the little bump that's coming out, it comes out in between two of them. So here's one, it's in between them. Here's one, and here's one, it's in between them. Here's one, and here's one, it's in between them. Normally for many of the centric diatoms, the round ones, these little things occur on the costae. And for this group, and this group alone, this genus, they occur between the costae. And so you can always tell, if you can see the costae, that you're looking at Discostella. And this is Astera costata. It's very large. It's slightly covered with junk, but I recognize it because it has a big hyaline area here in the middle, and then it has these very large sort of flower-like structures that are covered a little bit right now. But in 2020, I think 20 or 2021, we wrote a paper about this as an invasive species. It's all over Indiana. It's all over everywhere the Wabash River connects up to the Missouri River, which connects up to the Mississippi River. In all of those basins, this thing is present. And what's interesting, or was interesting for us, is that it seems to follow the distribution of Asian carp. And so we think, because Asian carp was brought over from a place where this thing exists, that it was actually brought here inside the Asian carp as a gut passenger, and that some diatoms will pass through the guts of the fish and come out the other end and be fine. And so a fraction of them will be consumed and dissolved and eaten, but most fish have pretty simple stomachs, and so stuff can pass through it without actually being digested. And so some of those diatoms clearly passed through these fish and came out the other end and were fine, and they managed, or at least this is our story, they managed to basically then colonize some of these river systems. And the reason that we know that is we have a whole bunch of lake records where this thing doesn't exist at all, and then suddenly it appears. And the timing of its appearance coincides with the timing on the appearance of Asian carp. Its distribution in North America also corresponds more or less with the distribution of Asian carp. And to expand this even further, it's something called a disjunct population. In other words, we see this in Asia and a little bit in Russia. So it's pretty common in China and a little bit into Russia. And then it's nowhere else. You don't find it anywhere in between those places and North America. And not just like the western part of North America, which you might think if it migrated or something, but rather you find it in these areas as a common species in places like China and Russia, and then it's common in Indiana and nowhere in between. And it's the same species. So like genetically the same species as well as morphologically the same species. So that's actually kind of a really interesting issue, where we can track the invasiveness by the fact that the organism basically doesn't exist anywhere in between, right? So like conceptually, how do you get a diatom from Asia to not just North America, because that's possible, but like a little part of North America that's connected to the river systems. And then all of these river systems, the Wabash and the Missouri and the Mississippi, people will catch fish from them and use them to stock little ponds. So this one actually came from Deming Park, which is a park that's nearby here in Terre Haute, and Maple Park, Dobbs Park. All the parks in the surrounding area are stocked with fish that come out of the Wabash River, and we believe that not only were these diatoms brought from the Wabash River, into the Wabash River from someplace, but that all of the lakes around the Wabash River, these little places where people stock them, they've now migrated into those systems and have become common diatoms in those systems. So this one's from Deming Park. The other ones that we have are from Dobbs Park. We found them in all these lakes that are basically adjacent to the Wabash River up here in around Terre Haute. So they are common. They can also be transmitted on things like people. So people might move a boat from one place to another, or on the legs of birds. Like that's a way that you could distribute it. Or people could get them on their shoes and go from one place to the other. But the problem is, Deming Park, you're not allowed to wade into it or swim in it. It's too small. So it can't be people, or like boats don't go in there. It's way too small for that. It could still be birds. It could be birds go from one place to the next, and then diatoms get caught on the wings or something like that. But so that's our theory or hypothesis for how that works. So you can ask, do invasive diatoms have detrimental effects on the ecosystem? Probably not. There's still diatoms, so they just generate oxygen and they're food source for things like fish, so they're not actually problematic in any way. This is Deming Park and Discostella asteriscata. So it's probably not problematic. They may be displacing local algae, like local diatoms, but I mean, from a conceptual point, does it matter whether I have this diatom or a different diatom? They basically fill the same niche in terms of the environment. They're food for fish and snails and other things. So probably it's not that big of a deal. We weren't trying to make a case that it was something terrible, but rather something that people probably weren't aware had happened. People don't care about algae. Generally speaking, anything that's small, that's this small, like diatoms, people just ignore. If it was really creating a problem, like it was an invasive species and it was creating nuisance blooms or something, or toxins, then people would care about it. But generally speaking, people typically don't care about algae because they view it as it's too small to care about. People care about fish because they can eat fish, and it's a sport. So if it's an invasive fish like Asian carp, then people do care about it. But if you can link the behavior of, we care about fish to, hey, we're also bringing these other passengers into the environment that are modifying the environment. They're not, they are in that system. But it's hard to say whether they actually are creating something deleterious to the environment or creating a problem. Yeah, it does count as... So we usually use the word non-native to avoid the sense of like we're being attacked by whatever. I use the word attack. But, yeah, really they are species that were brought in. If they really did piggyback on the... So, we don't know for sure that they've piggybacked on the Asian carp. We just have a lot of circumstantial evidence that suggests that they did. The timing of their arrival in all of the lake systems that we looked at dates pretty closely with introduction of Asian carp. And then the distribution also seems, both the modern distribution and the disjunct populations all sort of point towards it. But so if they weren't brought in by people in the Asian carp, then they are truly an invasive species. But even if they aren't, they're still a non-native species, right? They're still potentially being brought in by into an environment they didn't exist in before. What are you researching currently? Is work in progress work found on ResearchGate? No. I usually don't put anything out until I've published it. Right now I have a bunch of projects in Lake Tanganyika that we're working on. My work with Laura is on June Lake and Gull Lake. So Laura has been on the stream many, many times. She's a master's student here. So that's a bunch of modern research in the Sierra Nevadas, and then I have a bunch of undergraduate projects, a bunch of taxonomy projects. I'm playing hooky from a manuscript I need to actually finish that's going to describe 5 new species of Afrosimbella. So I'd like to get that in before the end of the week. But it's been reviewed and it's accepted with revisions, so I'm just making the revisions, and it's very likely to be accepted as soon as I turn it in. I don't think that anybody's going to complain about the changes. They were mostly technical changes, so that's good. But there's a lot of projects that I'm working on simultaneously, and then every grad student I have is also, their project is kind of my project. So you dislike algae. They tend to take oxygen out of the water for your animals. That is the exact opposite of what algae do, goat farmer Bluesy. What you hate are bacteria. Bacteria take oxygen out of the water. Algae add oxygen to the water. It's just that when the algae die, their decomposition leads to the subtraction of oxygen from the environment. Algae add oxygen to environments. That's what algae do. They're photosynthesizers. So your hate is misplaced. Wait, there's a hug? What's going on? Oh, they're just exclamation points. You dislike dead algae. That's fair. That's a fair statement. I will let that go. But what you really should hate is, let's see, what are they taking oxygen from? It's oxygen out of the water for your animals. I'm trying to think if the problem is really you have too many nutrients in the water, and so the algae grow in that system, and their eutrophication is a response of too many nutrients in the system. So I don't know who's adding the nutrients to your system, but I think maybe they're responsible, whoever that might be. This is Diatom amphora. It's the genus is amphora, and this species may be pediculus or inariensis. I don't remember which. I think maybe it's inariensis. I don't think the dead algae have any feelings about Bluesy. I think they probably just view themselves as being thankful that they had a place to grow and somebody gave them food. Yeah, we were just talking about how people care about animals and not algae or small things. Exactly. It's your hatred is misplaced, is all I'm gonna say. They're just doing their thing, which is growing where you give them food and they have water, and replicating and generating oxygen. So largely beneficial to the environment. But it's the bacteria that are the ones that come along and eat the dead or decaying algae. But I actually don't think you should hate them either, because all they're doing is remineralizing a bunch of the nutrients that are available in those organisms and making them available for another generation of organisms. So even that's probably not something to be, you know, hateful about, because if we didn't have decomposers, we'd be in a really bad spot. We need decomposers, and in water especially, because they help recycle nutrients in places where nutrients aren't very high. So, hate people who fertilize their lawns too much. Yeah, well, it could be their lawns. I mean, they're just trying to grow a lawn, so, you know, I think maybe we'd just do better without hating anything, and maybe just recognizing things are trying to live in the environment. You know, put your effort into caring for the environment rather than hating things that live in it. Yeah, exactly. Diatoms do not die. That's Barbados. That's actually part of the irony in my name. I don't know if that was obvious to people, but you know, I named myself after like Godzilla movies where you had a giant monster that was like attacking, and diatoms are tiny, right? It's like a 50-foot diatom or something. But they're also benevolent, which I think makes it extra funny, you know. So to me, my name is pretty hilarious. But I noticed actually one time I saw I Googled diatoms attack once as a phrase after I'd already named myself, and I saw somebody used it in a news article once. So in Fora, there's a bunch of little Fragillaria colonies all around in this sample, and I'm picking out the things that aren't that. So excuse me for like ignoring the primary thing in the field of view so that I can image all of the other things in the field of view. This is Melisira. That other one was Melisira. Pretty simple looking diatom in the light microscope. Relatively complicated looking organism when you start looking at them in the SEM. They have a bunch of little spines and they make these colonies. They're indicators of flowing water, typically. That's Melisira varians, which is a common diatom in lake systems and, in river systems. But this is another example of probably material being imported into the lake, because that probably is not something... There's no high energy environments in the park at Deming Pond. Although there is a fountain that oxidizes that system. So I suppose it could be related to that. Here's an internal view of the Discostella astericostata that we were looking at the beginning. We looked at the invasive species or non-native species I was talking about. It's actually a pretty diatom. If you're using the follower emotes, it does look like a button. I agree. Or a wagon wheel, or in fact, we have a fun game we play where I try with my lab. I try to get them to see things in like a hubcap, right? See things in nature, in the world around them, and recognize what diatom it is. You know, like what diatom is this? So if you saw a hubcap that looked like this, you could be like, oh, I know that one. But usually they're like patterns in things like curtains or pillows or art that you find at a hotel, maybe, or something like that. You often find patterns that look like diatoms. Or I do. You see enough of them and you start to see them in everything. Hey, Boba! Hello! Welcome in. Dislike results. Yeah, that's actually a fair way of thinking about it. You named your channel after cyanophytes because I thought it was cool how they influence our planet. Yeah. Marshmallow. They do look a little like marshmallows. This one does look like a button. Have I seen Mentango? It's the giant mushroom that attacked. Yeah, I know this movie because Invisible Dimensions, in my class, I teach a geology class called SEDSTRAT, and I have a final project for the students, and the sites that I give them, I used fake names for. like I want them to recognize that everything was completely made up, right? So the names of the sites where the samples were collected for my fake project for them are all horror movies and things from like Monsters That Attack and like Cthulhu references. So I have a whole bunch of really weird things, but Mantango is one of the sites in that project that they do, and nobody ever gets it. Every year I put these like because I think what could be funnier than having like horror movies as the sites for their final project in the names. But this is my sense of humor, to bury something that nobody will know right in front of them, and then they all know. It's funny because they're all standing up in front of them talking about like Mantango and like all these. Oh, one of the lakes is really obvious though. It's called Sharktopus Lake or something like that, where the sample is Sharktopus something. So you would think that they would know what Sharktopus is, if nothing else. But as a connoisseur of attacking monster movies, yes, I do know Mantango. When I was a kid, I thought bruises on your legs looked like buttons, and you would Jesus, Moosey. It definitely looks like a hubcap. Yeah, there's this this follower email. This one. Oh, I'm not logged in on my phone. Hang on. This one. This is Discostella astericostata right here. This one. Yeah, you found it. You wouldn't press them hard. No, I don't think anybody gets. I didn't use Sharknado. I used Sharktopus, but Sharknado would be a good one. Kids don't know the classics. Exactly. You were. That is science. That is a type of science for sure. This is an internal view of the diatom Discostella astericostata, and no doubt that that's what that is. Look how cool it looks. That's a cool looking diatom. See, costi, and then the Mantophotoportula is between the costi. Here's a costi, Mantophotoportula is between them. So you can tell. You're learning diatoms slowly by watching my channel. You'll see something, eventually it'll click, and you'll be like, I know that one. It's a consequence of having the names thrown at you. They start to sound like, oh, I know that. I've heard that word before. And then eventually it's like, they're old friends. Hopefully. All these diatoms that we're seeing here in the foreground are Fragillaria, I don't know, Mesolepta maybe? There's sort of a moderate nutrient indicator, and then these capsule-shaped things are Melisira. That's a Cockanice down there, and that is Reucosphenia, which I think I saw a Reucosphenia yesterday as well when I just glanced through these really quickly to try to see what was here. But Reucosphenia is another flowing water indicator, which seems to suggest possibility that these were stocked pond diatoms. Here's a really long ulnaria, which I'll try to image, seeing how it's more or less already at the right angle. This is the center bit. There. Now it's in focus. So these little guys are Fragillaria. Like I said, probably Mesolepta. They live in colonies. They're sort of like a moderate nutrient indicator. And then the rest of these things are flowing water indicators, or this lives in metaphyton. They can live attached lifestyles, but I think this one is a metaphyton type. You can see how big it is relative to the rest of those. The length of this thing is around 200 microns. So it's about 5%. No, wait. 200. It's 20% of a millimeter. Yeah. So if we stack a bunch of them together, we could almost make a millimeter. It was required viewing in your cyberpunk course. Oh, that's pretty cool. They have courses on cyberpunk? Man. I never had anything cool like that when I was a kid. That right one looks like a Bond villain. Oh, it's got spines that interlock, yeah. It does look a little bit like Jaws, like a shark jaw. A bunch is 100. Is that a technical term? I didn't realize a bunch was a technical term. It has a specific meaning. 144. I'll have to look that up. I thought a bunch would just meant there were a whole lot of them. We would need 5 of them to make up a millimeter. All right, I trust you. I'm So this is Melisira. There's a hole valve and there's a hole frustule and there's a hole frustule. They're joined together. There's a girdle band wrapped around both of them here. And then all of these are little colonial guys. Not super interesting. That they're basically all the same species of Fragillaria, probably Mesolepta or something like that. I haven't looked closely at them to tell. But they have an indentation in the middle. And the one that we're looking at right now is Ulnaria. This is Ulnaria. If I can spell it correctly. I'll throw that up there on the screen for you. They look like that. And then there's Fragillaria. That are closely related. And did my little pop-up thing not work? Pop-up thing. It's because it was already open, I think. There's Fragillaria. You can see they live in little clumps or clusters and attach face to face and create ribbon-shaped colonies. They have little tiny spines that interlock that hold them together. And then there's probably a couple species of Fragillaria in here. I didn't look closely. I'm not that focused on what the Fragillaria are, because there's a bunch of them, and they probably mean the same thing. Just like relatively high nutrients, but not super high nutrients. Okay, this is Ulnaria. I'm just trying to find some diversity of things. And I think that there's a Gumphanema hiding in the background. Let's see if I can find anything else that's here that's obvious. You can see how many of the Fragillaria there are. There are overwhelming numbers of them. This is Astera costata again. This one's actually better preserved. There it is. This is our little invasive diatom that I was talking about, Discostella Astera costata. And very distinct looking. And it's weird how they have all these shapes but do the same thing. Well, they don't do the same thing. That's a little bit of a... the shape actually does play a role in the organisms themselves, which is to say, this has a different shape. It actually has like a one side concave, and it has a raphe, and it lives an attached lifestyle. And this one, the round one, floats in the water, in deep water. This long skinny one usually lived in little tufts or in metaphyton, and these ones live in intermediate water depths. So, not exactly the same things. Similar things, I would agree. But they are different habitats that they live in, subhabitats within the environment. Okay, I'm going to image this. This is a Diatoms. It's Confervasa, I think. I think it's still Diatoms. And then I think we're going to end relatively early today. But they do have some sort of structural aspect that's linked together with their function. So it's not exclusively like they all live in the water, but they live in different parts of the water. So, okay. So this was just a little short stream to try to get some work done and look at some cool things, which I think we've accomplished. I think we're going to raid out into my friend Estrella is streaming, and so we're going to go give her a visit. I think she's making stuff out of clay. A bunch of bananas is about 100. Is that true? Okay. Okay, well, we're gonna raid Estrella, and so she's doing some cool art stream. She's I think maybe she's trying to draw today, but usually she does digital art or drawing, or she's making stuff out of clay. So the rest of my friends that are on that are doing stuff are exercising, and I feel really weird about like raiding into somebody's channel when they're exercising, so we're not gonna do that. But thanks everybody for hanging out, and I'm going to throw this thing ending screen up right here. And I don't know, we got some people that hang out and ask interesting questions today. It was a relatively short little stream. We got subscription from Evan.pro, so thank you, Evan, if you're still around, for hopping in and giving the subscription. And everybody else, just thanks for hanging out and visiting with me. And Lil Chook's going to be happy because we're going probably to combine her tabs to into one, from mine and Estrella's down into just one. So, oh, thank you. Yeah, I do birding streams probably on the weekend and maybe a little bit during the week in the next couple of weeks. We'll see what happens to come by my backyard. So, all right, thanks everybody, and we'll catch you next time. Tomorrow, probably.